Welcome back to Exploring the Metaphysical with One Foundation. What you're witnessing here is an interactive exploration of one of biology's most fundamental processes, cell division. Through this parametric system, we can manipulate and visualize the complex three-dimensional structures that emerge during cellular reproduction, offering insights that traditional two-dimensional representations simply cannot provide. The model you see before you represents a dividing cell at macro scale, allowing us to examine the intricate geometric patterns that govern how life perpetuates itself. Each adjustment to our parameters reveals different aspects of the division process, from the initial stages of DNA replication during interphase to the dramatic restructuring that occurs during mitosis or meiosis. As we rotate and examine this structure, notice how the cellular geometry transforms. The orange filamentous networks you observe represent the dynamic cytoskeletal elements that orchestrate division. These aren't static structures but rather active participants in the choreographed dance of cellular reproduction. The precision of these models is remarkable, with some containing over 100,000 vertices, capturing the minute details of protein interactions and membrane dynamics that occur at the molecular level. Through our parametric controls, we can simulate different phases of the cell cycle. Watch as we transition from the growth phase of interphase, where the cell accumulates the resources necessary for division, into the dramatic reorganization of mitosis. The spherical formations you see emerging represent different configurations the cell adopts as chromosomes align and separate, ensuring each daughter cell receives an identical genetic complement. The beauty of this interactive system lies in its ability to generate alternative geometries that reflect the diversity of cellular division strategies found throughout biology. Each parameter adjustment reveals new possibilities, new configurations that cells might adopt under different conditions or in different species. The white wireframe structures in the background represent simplified versions of these complex forms, allowing us to understand the underlying mathematical principles that govern cellular architecture. As we explore these models, we're not just observing static representations but engaging with the fundamental algorithms of life itself. Surface tension, protein dynamics, and genetic regulation all contribute to the final three-dimensional forms we observe. The parametric nature of this system allows us to ask what-if questions about cellular division. What happens when we alter the timing of chromosome condensation? How do changes in cellular volume affect the division process? These explorations help us understand not just how cells divide, but why they divide in the specific ways they do. Most importantly, we need to ask ourselves, is our representation of cell division precise? Will the scientific and medical field recognize our accomplishment? Only time will tell. Until then, continue to follow us on our journey to reshape the world by shaping our reality.